This is a slightly longer version of a talk presented on October 31st, 2022 to the Hitchhiker's Advanced Guide to Quantum Collapse regarding quantum collapse associated with electron tunneling in substantia nigra pars compacta, or SNC tissue. First, I'll be discussing evidence of electron tunneling in ferritin, and then I will discuss evidence pertaining to the ferritin structures that are in SNC tissue. And finally, I'll talk about modeling electron transport in the SNC and the associated wave function collapse. Ferritin is a spherical iron storage protein. It's formed from 24 subunits that can be light chain, heavy chain, or mitochondrial subunits. It can store up to 4,500 iron atoms, primarily as Fe3+ in ferrohydrite form, but it can also form magnetite or ferrohydrite precursors, and other substances can also be stored inside of the ferritin. Electron tunneling behavior was first observed in 1995 at low temperatures associated with ferritin, and then in 2005, room temperature tunnel for microscopy. Professor Christian Neuhaus is one of the leading experts in testing electron tunneling in ferritin, and one of the things that he tested was coherent tunneling as a function of iron loading. He used this ferritin-based junction to avoid forming the ferritin core, which was a concern with the conductive atomic force microscopy testing. He found that the size of the ferritin particles increases with loading, as does the core contents. But regardless, the type of tunneling that he observed was coherent tunneling, which is to say that it was temperature independent. And for ferritin particles, 8 nanometers or so, he observed individual coherent tunneling events and then sequential tunneling events for ferritin particles about 8 nanometers and larger. Subsequent work performed by Professor Neuhaus showed that the activation energy of ferritin changes as a function of iron loading, with higher activation energy for greater iron loading. It also demonstrated that the tunneling process is not strictly coherent with the increased iron loading, but includes some incoherent tunneling. Professor Neuhaus also tested the effect of magnetic fields on tunneling through ferritin, and proposes that long-range tunneling is assisted by magnon excitations of ferritin. The magnons are created in conjunction with light that can form inside of the core of the ferritin under certain conditions. He also discovered that the type of tunneling is a function of the junction material or contact material associated with the ferritin. In this test, he looked at bilayer graphene and showed that for apoferritin, or ferritin that has no iron in the core, the tunneling behavior was different from low iron ferritin and high iron ferritin, and also that the Fermi level of the bilayer graphene changed as a function of the iron loading. What this indicates is that in vivo, the substances in contact with the ferritin and the tissue structures that the ferritin might be disposed in will have some potential impact on tunneling behavior. Electron spin characteristics of ferritin have also been measured using nanodiamond nitrovacancy spin detectors. The relaxation time and spin coherence time were both reduced at room temperature, as can be seen in these graphs with the ferritin absorbed sensors on the left and the planes unabsorbed sensors on the right. Other interesting electrical characteristics of ferritin are its ability to store electrons for at least several hours. In this test, electrons are loaded onto the ferritin cores using electron beams from a linear accelerator and laser pulses. And a chelator was added over a period of time to different samples. And what was observed was that the iron release from the core is representative of Fe2 ions that are water soluble instead of the ferrohydrite Fe3 ions that are not water soluble. But that characteristic was consistent depending or independent of when the chelator was added. What was not addressed in the study is how many electrons are stored, particularly how many electrons might be stored in vivo during normal biological functions that involve ferritin. Ferritin has also been tested for electron transport over long distances. In this study by Professor McCavier, she showed that disordered layers that were deposited in a layer by layer fashion were capable of conducting electrons through sequential tunneling over a distance of 40 microns at current level 0.3 microamperes at 3 volts applied with this current voltage behavior. That was also observed subsequently in tests I had conducted in conjunction with Professor Kai Shen at the Ningbo Institute of Materials, Technology, and Engineering. We had 32 different dyes, tested a number of different configurations and combinations of layers. The maximum current we measured was 3 microamperes across an 80 micron gap at I note that 75% of those dyes did not result in any appreciable or measurable current being observed, which shows that this disordered layer by layer process is not consistent and does not consistently result in structures that have sufficient order to support sequential electron tunneling over long distances. But there were at least 25% of the dyes did and similar current voltage characteristics. Professor Middleton, one of the pioneers in studying transport through quantum dot arrays, did a study which used a simplified analysis treating quantum dots as metal islands separated by capacitances with the wave functions localized to a single dot and other simplifying assumptions. He looked at a 2D disordered array and observed similar current voltage behavior with a linear response that had a knee and then further linear response with a transition voltages around what was observed in the test with layers of ferritin. So this is not a direct model of what was tested, but it's a similar model and it has similar results. I note that he had a temperature effect at 0 degrees to eliminate the effect of tunneling probability from his analysis, which is not a factor with the ferritin because ferritin has been observed to have room temperature tunneling. But this indicates that the earlier measurements of tunneling through ferritin arrays are consistent with models. The test performed by Dr. Shen, we're using a 4-terminal device that I designed to determine whether switching or routing could be occurring in these disordered ferritin arrays. We tested different configurations. This one die, we observed this very clear nonlinear switching type behavior, which is essentially electrons flowing from the left side to the right side, but not back. This one particular test involved electrons flowing through all 3 grounded electrodes, V1, V2, and V3 grounded from this electrode, but the return path did not have appreciable current. I have a $10,000 here was the cost for these tests. I'm a citizen scientist and self-funded these tests. I think this indicates that additional testing could be performed if there's an interest to further investigate this switching or routing behavior. The switching routing behavior appears to be due to a Coulomb blockade, which is a quantum dot type electrical property. Because the disordered arrays has random configuration, some of the dyes would appear to have had constricted areas similar to this, closer to one electrode than the other. The iron in the core is typically at a +3 oxidation state, such that when electrons are injected from one terminal near the narrow end of the constriction, the iron is reduced to +2 oxidation state. There are cores adjacent that have iron in the +3 oxidation state for the electrons to tunnel to, so that the electrons can flow in this direction. But flowing in the direction results in these oxidation states being filled and creating this Coulomb blockade that requires certain levels of voltage to overcome the blockade, which is also observed in other dyes. And I note that after these test results were published, Professor van der Zand did some tests, individual ferritin cores at 4 degrees Kelvin between 2 electrodes using single electrons, and found excellent agreement between his experimental data and the Coulomb blockade theory. So this evidence indicates that ferritin has at least some behavior that is quantum dot-like in terms of being a well, isolation well for the electrons to tunnel between, consistent with the way quantum dots function. Even though ferritin is not a quantum dot, is much more complicated than a quantum dot with the protein shell structure and the ferrohydrite and magnetite core contacts. So now I'll discuss evidence of ferritin structures in SNC tissue, similar to the ones that were tested in the disordered layer devices. The SNC is a nuclei in the basal ganglia that's associated with action selection. It has a large number of dopamine neurons, but a small percentage of them are very large, such as this one here, which has been stained to show neuromelanin and is about 20 to 30 microns in diameter. These large neurons all group together in a relatively small area and are closely disposed adjacent to each other in areas that have iron and ferritin in particular, as I'll show. Inside of the dopamine neurons are neuromelanin organelles. Neuromelanin is typically a 30 nanometer spherical pi conjugated polymer, which behaves like an N-type semiconductor material. Ferritin, with its ferrohydrite core and other Fe3 plus type iron contents, behaves like a P-type semiconductor material. So this is an interesting configuration. In this electron microscope photograph, you can see small black dots that appear to be layers around this neuromelanin organelle. It's not possible to determine if those dots are ferritin except through the use of something like immunogold markers. I'm sorry. These immunogold markers are tagged with anti-ferritin, so they're attracted to ferritin, and you can see at least some small number of them found their way into this structure. Obviously, there's not a one-to-one match. There'd be an enormous number of immunogold markers, but this was indication that there is ferritin outside of these neuromelanin organelles. More recently, electron spectroscopy was used to identify iron in layers outside of neuromelanin organelles. most of the iron in the SNC is stored in ferritin. So this is a good indication that there are layers of ferritin outside of these neuromelanin organelles. This particle-induced X-ray imaging also shows iron between large dopamine neurons in the SNC. So this is an indication that there's iron between those large dopamine neurons. This can be seen in this electron microscope photograph of a glial cell from the SNC. The paper does not describe these black dots as being ferritin, but they are very similar in appearance to these black dots in human placental tissue, which was identified to be ferritin. Dr. Michalik was testing the human placental tissue for increased magnetic ordering using small angle neutron scattering. And she did find that there was substantial increase in this magnetic ordering compared to the ferritin after it had been removed from the tissue and tested separately. This is consistent with prior observations that small angle neutron scattering provides insights into quantum dot systems and quantum dot solids, and this increased magnetic ordering is an indication of a quantum dot solid type behavior, again with ferritin, which is not a quantum dot, but behaves like a quantum dot. Professor McCott at the University of New Mexico tested ferritin cores after burning the protein off. So this is just the ferrohydrite core and other materials. It's disposed on silicon, a relative degree of order than the disordered array tissue. And Professor McCott also measured increased magnetic ordering in response to small angle neutron scattering. So this evidence shows that the tissue associated results are consistent with silicon device results, and that ferritin is probably behaving consistently in either environment. So in order to see if this testing was electron behavior was occurring in the substantia nigra pars compacta tissue because of the structures that were observed there, I spent an additional $4,300, self-funded these tests. I ordered fixed substantia nigra pars compacta tissue in 5-micron pieces that were deposited on aluminum foil from AMSBio, which is a tissue research supply house. They shipped the tissue directly to EAG Labs in Silicon Valley, which has expertise in conductive atomic force microscopy testing. The area that was tested was a control sample in this scan, which was several millimeters away from the area that had these large dopamine neuron cell body structures. Conductive atomic force microscopy testing uses a nanoscale cantilever that bounces along the surface of the test sample in a line and takes 500 current measurements along the line, and then it does 500 lines in a scan for a total of about 250,000 current measurements. This sample scan was about an 80 micron square area, again in a part of the thick slice of tissue that did not have neuromelanin or ferritin, but which did have thin areas. You can see the negative currents here, which would be in the correspond to these thin areas, where a convention of electrons flowing from the probe with a -10 volt bias to ground was negative current. There were also areas where there was insulation provided by the tissue, even with field strengths of up to 2 million volts per meter. So that's expected insulating type behavior of this tissue and conduction of currents to ground in thin spots. When we moved the testing over to the neuromelanin organelles, though, with ferritin structures, a quite different current response was measured in those 250,000 measurements, with widespread positive currents This is a zero current line, so there's these large positive currents observed. That's essentially electrons flowing from ground into a negative biased probe tip, which is an electron source, which doesn't make sense from a conventional standpoint, but from a quantum tunneling standpoint, makes sense. The ferritin and neuromelanin structures are near the top layer and are isolated from the ground plane. So tunneling electrons would create a more electronegative environment than the probe would push electrons up into the probe tip. To get higher resolution, we decrease the scan area to 2 microns. So that's a 2 micron square with 500 measurements and 500 rows, and we could see pretty clear structures that appear to correlate to layers of ferritin outside of neuromelanin organelles. In this sketch, I've shown equipotential lines to demonstrate how the oscillating probe could create a dV/dt that could cause electrons stored in the ferritin cores, or electrons associated with pi conjugated polymers in the neuromelanin organelles to gain energy and tunnel. It's a very transient current measurement. It changed over time, a very short period of time when the probe was started to touch the surface, and then during the entire period when it was in contact at the peak, and then there's a third measurement taken over the entire oscillation period, which was much less. So with this evidence of electron transport in SNC tissue, the question follows of, is there electron wave function collapse occurring, and to what extent? And this could be modeled and potentially measured too. So it's generally accepted that when you have two quantum dot structures or wells, Coulomb wells, and there's a tunneling event, an electron moves from one to the other, that the electron becomes coherent in order to tunnel, and then the wave function, the coherent wave function, collapses well, or quantum dot. And this was particularly interesting, this paper by Stodolsky, where he conceded that even though he prefers the amplitude point of view, that he could arrive at conclusions about movements of quantum dot electrons by collapsing the wave function. And this was interesting because Stodolsky apparently believes that there is no wave function, but I guess what he's saying is there is a wave function, and collapsing it could explain this too. But I think it's generally accepted that collapsing the wave function occurs. Now, quantum dots are different from ferritin. So when you talk about collapsing the wave function at ferritin, the question is, what is it collapsing on? Is it collapsing on iron, Fe3 plus iron in the core, presumably? How is that occurring? What happens to the electron after it collapses? Because the Fe3 plus iron is in a ferrohydrite crystalline structure, and all of a sudden it becomes Fe2 plus iron is reduced, and it no longer can maintain its structure in the middle of the crystal. So, and then you have a bunch of other components. So it's a very unusual chemical process going on, and how the electrons can be stored for hours within the core if they bounce around from crystalline structure to get trapped in other configurations. So that would have to be modeled. In order to model that using existing packages, software modeling packages, first we have to make sure that we have the entire environment correctly modeled, and that environment would include the cell bodies, or soma, which have an associated axon with an impedance is determined by the membrane potential, and then the ferritin cores between cell bodies. The source of the electrons would be from triplet electron generation from dopamine metabolism, which generates quite a lot of these triplet electrons. And then you would have to look at how the electrons are tunneling through the soma with the neuromelanin organelles and ferritin structures, and then through the glial cells. Between them, the membrane potential elevated from the rest potential of minus 70 millivolts to a higher potential like minus 30 or minus 20 millivolts. That less electronegative membrane potential would not repel electrons as well as the more electronegative membrane, so the electrons would essentially be pushed to the more active axon. And that's consistent with two different lines of study. One is on predictive reward signaling of dopamine neurons, and then one is on action selection. Professor Schultz found that dopamine neurons are activated by rewarding events that are better than predicted, remain uninfluenced by events that are predicted, and are depressed by events that are predicted. And that's using this structure where cortical inputs that signal when events are better than predicted and then don't signal when events are worse than predicted are being received at the striatal neuron boutons on the dendrites, where the dopamine varicosities from the axons of the dopamine neurons are connecting. So this is a very unusual structure. Usually a neuron has excitatory inputs that are received at dendrites, and those cause action potential to be generated in the cell body that drives neurotransmitters through the axon. But this structure appears to go the other way, where the cortical neurons that are coming in and received at the axon are causing these neurons to fire. More recently, Professor Pascal Kaiser at Harvard also observed that for movement control, dopamine modulates moment-by-moment activity in the striatum to mediate action selection. So again, this is consistent with this structure and the cortical inputs causing the dopamine neurons to fire and being associated with striatal neurons firing, in this case for action selection. So this entire system could be modeled using existing packages. Just as a little additional neuroscience, this is an example of the axonal arbors associated with those large dopamine neurons. They're very extensive, and these are unlike most dopamine neurons that do not have these small arbors. It's maybe 1 to 2% of the total population of dopamine neurons in the brain are this large. There are cortical axon connections where all the axons connect to the striatal dendrites. Throughout here, if you have a lot of these neurons in parallel, some mechanism would be needed to prevent them all from firing simultaneously, which could cause seizures or prevent predictive reward signaling learning. And I note that ferritin structure is that mechanism. It could integrate these cortical signals even when no action is being selected. So the cortical signals could be coming in. There's no action selection being generated by cognitive processing, and then this integration would integrate all the information that's being received by the structure. But going back to modeling this structure using computer modeling software, PYXI is one package that could potentially be used to model the electron tunneling. It allows for modeling of quantum dots, which would be similar to the ferritin. It models decoherence as the electron wave function collapse to adiabatic states. So this would be the electrons tunnel to different ferritin cores and collapse. And then somehow you would have to modify the model to show that those electrons subsequently become coherent through interactions with magnons in magnetite or in other manners and tunnel to the next ferritin core over. This package may be limited to several hundred atoms, and the ability to model magnons or electron spin is unclear, but it could probably be modified. It's an open source package. There's also the NEMO5 package. NEMO is a class or a family of packages. NEMO5 is the most current. It seems it could be used to model very simple SNC neuron structure. In this example, they modeled a single electron transistor that's used for selective tunneling of an up or down spin electron to these quantum dots. So it can definitely handle spin. This mesh has 550,000 nodes and 3.5 million elements, which is a rather large model, but it has different materials and a lot of different physical structures. So it may be possible to use this package to create a model that is close to what's going on in the SNC. The models could be matched to tests using quantum dots that are tagged to be connected to ferritin using anti-ferritin. So basically, this paper shows a quantum dot fluorophore that's used to identify ferritin using a UV excitation source. But if there is electron tunneling associated with ferritin, if ferritin is storing electrons in the core and those electrons are becoming coherent and can tunnel, then they could tunnel to a quantum dot that's attached to the exterior shell of the ferritin and generate photons. It's similar to the mechanism that's used to generate photons in quantum dot LEDs. So by using these quantum dot fluorophores without the UV excitation source, it could potentially locate photon generation associated with tunneling in live tissue with ferritin structures. So in summary, it appears that ferritin supports coherent electron tunneling, and that this would probably occur in vivo. There's no reason it would not occur in vivo. By all indications, there's evidence that it does occur in vivo. Each tunneling event would correspond to a collapse event. And because there's so much ferritin and so much tunneling going on, you're talking about a very large number of collapse events continuously occurring as these electrons tunnel around through the SNC neuron structures. This electron transport between the large SNC neurons through those ferritin arrays and the associated wave function collapse could be modeled and tested. So thank you for listening to this presentation. And if you have any questions, please contact me at your convenience. I am happy to answer questions about this very interesting and new area of study.